Thanks for the invitation to be here. It's a pleasure. I'm really not going to focus too much on chromosomes because I know I have Joris following who's going to do a fantastic job on that. So I was asked really to talk about karyomapping, and karyomapping really means preimplantation genetic diagnosis rather than screening. I guess just to begin with maybe a definition of that. I mean, when we're talking about PGD, what we're really talking about is looking at a sample from an embryo that's been generated using in vitro fertilization technology, doing a genetic test on that sample in order to find out whether or not the embryo has been affected by a particular inherited condition. And this is used not as a screening method, but for families who already know they're at very high risk of transmitting a particular disorder. So it's very specified. We're looking at one particular thing. So really, when we're talking about PGD, we're talking about inherited conditions rather than PGS when we're talking about looking at genetics again, but in order to improve IVF success rates by identifying the most viable embryos. Now, you could consider PGD to essentially be an alternative to traditional prenatal testing for these couples who are already known to be at high risk. So of course in the traditional route they would probably become pregnant naturally. They would test the pregnancy using chorionic villus sampling or amniocentesis. But of course the downside of that is if you find that the pregnancy is affected, you really don't have any pleasant options. You're going to either need to think about a termination or continuing with the pregnancy knowing that the child will be affected. So the way that the PGD has traditionally worked is you have an IVF cycle. This is regardless of whether the patients are fertile or not, because you're going to have to get access to pre-implantation embryos. So that of course involves ovarian stimulation. Hopefully multiple eggs will be generated. They're going to be fertilized with the partner's sperm to hopefully produce several embryos. A biopsy is going to be performed of those embryos in order to get some material for testing and hopefully find some that are unaffected, which would then be transferred to ultimately give a healthy pregnancy and a healthy birth. Now, PGD over the years has been done within a fresh IVF cycle, so it's by necessity had to be a very rapid process. Of course, we usually refer to the day of egg collection as day 0. Fertilizations happen by day 1. The biopsy of the embryo, the removal of material for testing, either on day 3 traditionally or increasingly these days done on day 5 at the blastocyst stage, and then a transfer, very rapid genetic test. I mean, this pushes the genetic technologies rather than like in traditional clinical genetics where you may have a couple of weeks to get a result and literally tens of thousands of cells. Here you often only have one cell and maybe just 24 hours to get a result. Increasingly though, these days people are vitrifying, freezing, cryopreserving the embryos after biopsy. So that at least removes some of the time pressure that we've had in the past. Of course, the main aim of PGD really is to avoid the whole issue of pregnancy termination. So you could ask, okay, so that's obviously desirable, but what particular patients do we get who go down the PGD route? Well, of course, there are those who have an opposition to the whole concept of pregnancy termination. This may be for their own personal ethical reasons, maybe for religious reasons. It may be because they've gone down the traditional diagnostic route of getting pregnant naturally and having a prenatal test, but they've had a negative experience of it. Maybe they've had affected pregnancies, they've had terminations, and they don't feel that they can continue down that route again. Interestingly, many of the patients, in fact the majority that we receive for PGD, although they're having IVF, they are actually perfectly fertile. The other kind of patients that we get are those that are sort of doubly unlucky. They're unlucky enough to be infertile and need IVF to conceive, and they're unlucky enough to be in a family where an inherited disorder is being transmitted. Now, of course, for them, it doesn't make sense to sometimes struggle for years with IVF to finally achieve a pregnancy, find it's affected, and then have to think about terminating that much wanted pregnancy. For them, of course, it makes great sense to look in the embryos. There's various different times you could test, as I've already mentioned. You could look at polar bodies taken from the egg. That's quite a nice approach, although of course it will only tell you about the maternal contribution to the genetics of the embryo. The most widely used method for really the last 25 years has been to take a single cell at the cleavage stage, so blastomere biopsy, But really over the last 5 years or so, there's been a huge shift towards biopsy of usually about 5 cells from the trophectoderm of the blastocyst stage embryo. So we're talking maybe day 5 there. The material is removed, it's taken away for testing, and hopefully we'll find an unaffected embryo. This graph here just shows an indication of how we've seen this shift towards blastocyst biopsy. So you can see back in 2008, this is just from the Reprogenetics lab, doing about 2,500 procedures per year, all of them with day 3 biopsy. But as you see, as we go through to 2015, there's a huge increase in the amount of embryo testing being done. It's going up by about 2,000 per year. This is number of cycles, not number of embryos. But you can see that the day 3 biopsies really fade away. So more than 95% of all embryo testing now is done in conjunction with trophectoderm biopsy. I can show you what one looks like. This is one that was done by Tracy Griffiths at Oxford Fertility a couple of years ago. What you can see here, a pipette, very magnified, just applying a suction to hold this embryo in place. It's at the blastocyst stage. You can see the zona pellucida quite nicely around the edge. And you can see this embryo is already beginning to hatch. If you looked at this embryo again a few hours later, what you would see is an empty zona pellucida and the free-floating embryo. This is an ideal time to catch it for biopsy. So let's see Tracy do her work. So just zooming in, she's now picking up some of these trophectoderm cells. You can just see the inner cell mass there. Embryo usually collapses when this happens, but not always. And then she's literally tugging at these cells, and every now and again you'll see the image flick slightly. That's as she's hitting cell junctions with a laser as well. So it's quite an invasive, quite brutal procedure where you're tearing and lasering cells in order to remove them. But the difference of this compared to a day 3 biopsy is that the day 3 embryo, there's the embryo, always looks a little bit sad afterwards, but it recovers very nicely. And they saw the little bit of tissue that had been taken off. So the day 3 embryo, you're only taking 1 cell, and it doesn't look as violent as that, but the day 3 embryo is infinitely more fragile than the blastocyst stage embryo. So although this seems a more invasive procedure and you're taking more cells, you're actually taking a smaller part of the total volume of the embryo, and it tolerates it much better. However, I would say, and I was just discussing this with Alan Thornhill this morning, I think in the sort of rush to move from day 3 biopsy, which has become a little bit discredited, towards day 5, I think there really has been, it's been brushed under the carpet that actually you can still do severe harm to a day 5 embryo in the biopsy procedure as well if it's not done properly. I think we always have to remember that this is a very skilled procedure and it shouldn't be done lightly. So even blastocyst biopsy may have a small impact. The other issue is that the biopsy is costly. You're going to have to get a very skilled embryologist to spend a significant amount of their time removing these cells very carefully from the embryo. You're also going to have to buy a laser and you're going to have to maintain that, and those are quite expensive pieces of kit. So there is an increasing interest in doing things like analysis of the culture medium. We find that there's actually a surprising amount of embryo DNA in the culture medium. Or potentially, here again we see a blastocyst, potentially going into the fluid-filled blastocele cavity and removing some of this fluid, which again we see, actually does this run? Oh, sorry, don't have the video for that one, I don't think. So you can actually suck out this fluid using an ICSI needle or other small pipette, and you do sometimes find that there's a small amount of DNA in there as well. So these are things that people are actively exploring at the moment. I think it's far too early to say whether this is ever going to give you sufficiently robust information for clinical use, but certainly you do sometimes find DNA in there. It's a comparatively easy procedure, minimally invasive or non-invasive if you're looking in the culture medium, and you certainly wouldn't need a laser. So anyway, to get back to the genetic diagnostics, really the first PGDs were done right at the end of the 80s, early 90s by Alan Handyside, was the first to really publish on this. First analysis was just looking at the sex of embryos in order to avoid X-linked disorders, but rapidly evolved on to being able to look at individual gene mutations. We've now done PGD for more than 300 different disorders, and really since the sequencing of the human genome, 15 odd years ago now, it's possible to do PGD for almost any inherited condition as long as the underlying genetic basis is understood. The diagnostics are, I think, considering the challenges of PGD, extremely accurate. They're typically more than 99% accurate. And we've seen a very rapidly growing demand for these over the last few years. These are just cases in Oxford, and you can see that they're going up quite quickly, up to about over 300 a year now. The traditional methodology looked something like this. You would do a biopsy, and it would have most often been done a single cell on the cleavage stage. That cell is going to be washed, it's going to be put in a PCR tube, and then an amplification of the DNA is going to happen. Of course, there's not enough DNA in a single cell to see it directly, so of course you're going to have to do some PCR to amplify it to a detectable level. And then you can go ahead and detect the mutation, and that will give you your diagnosis. But the problem that you face in single cell analysis is that of allele dropout. That's one of the biggest problems, I guess, along with contamination, that you need to face. It's a problem because it's relatively common, and essentially what it is, these are all cells from the same embryo. These are heterozygous, got allele A and allele B. You can see that's being detected in most of the cells, but there's one just here where we're only detecting allele B. This is allele dropout. It's where you have a heterozygous cell, but after PCR amplification, you only detect one of the two alleles. One of them just doesn't amplify for some reason. Of course, this can be very problematic for diagnostics. Imagine if allele A was actually a dominant mutation. Then this embryo is affected, but you're only detecting normal DNA, so you're going to misdiagnose it as a normal. Problematic because, as I say, it isn't rare. It's actually very common. And so what rapidly happened with the conventional methodologies was the appreciation that you could not rely just on detection of the mutation. You had to do more. And so people started to look also at linked polymorphisms. So for example, here's your chromosome, here's your mutant gene just here. As well as looking at that, we're going to look at maybe a polymorphism nearby it here, and maybe another one up here. And in fact, people tended to look at 5 or 10 individual polymorphisms very close to the gene causing the mutation. And that gave you a kind of backup analysis based on genetic linkage that would make your test very accurate. Just a cartoon just to illustrate that. Here's your mutation site again, and on the same chromosome, a polymorphism. Obviously, this person has 2 copies of the chromosome and 2 different versions of the polymorphism. You hope that you do your multiplex PCR and everything amplifies perfectly, in which case you'll see mutant and normal copies of the gene and the 2 polymorphisms, 2 alleles of the polymorphism. But if you get allele dropout so that you fail to detect your mutation, you can still infer that this mutant chromosome must be present because you're detecting this green version of the polymorphism here, which you know is inherited along with it. So that's how you get your backup. The kind of polymorphisms that were used for this are hypervariable in nature. They're the same kind of short tandem repeats that are used for DNA fingerprinting, in fact. And so you would also get a kind of basic fingerprint for the embryo, which was very useful for detecting the other great scourge of single cell diagnosis, which is contamination. Obviously, if you have one cell in your tube, if any other DNA gets in there, you're going to have run into a serious problem. So by getting a fingerprint, you could see, is there the fingerprint of one individual in the tube, or are there fingerprints of more than one individual, indicating contamination? That's just shown here again, a mother and a father, both heterozygous, but with different alleles, which we can colour in. They're going to have 4 possible genotypes in their embryos. So when we look at these link markers, we want to see one of these possible combinations. If we saw another peak out here, well, that's not from either of the parents, so it must be a contaminant. I'll just skip through that. So the PGD protocols using these kinds of conventional technologies were highly successful, but they did have some limitations. I guess we're in an area here of personalised medicine, and they were a very nice example of that. You would have a tailor-made protocol for almost every couple that came through the door. So that's a nice example of personalised medicine. But of course, that personalisation comes at a cost. There's often extended workup times. In our lab, when we're doing a conventional PGD protocol like this, it typically takes us about 3 months to develop it. But there are many other labs around the world where it may take a year or even slightly longer to develop a test for one couple. And that's extremely problematic. Partly, of course, because there's a diminishing chance of IVF success with advancing age. In some countries, PGD is covered by the national healthcare systems, but usually only up until a certain age. And if you begin working with a patient when they're near that threshold, by the time you've got the test ready, they may be beyond it and no longer be able to access funding. The final thing is that really PGD is a major undertaking for a couple, especially for a fertile couple, to opt to go through IVF with all its uncertainties and go down that roller coaster ride is a major decision. And when they've made that decision, they really want to get on with it. They don't want you to then tell them, okay, now wait 6 months. The other issue is that the protocols that we have, although very accurate at detecting the mutation, they don't look at anything else. They're very targeted. And one of the main things they miss are chromosome abnormalities. They don't detect aneuploidy. Now, I know Joris is going to talk about this far more, but I'll just touch upon why we care about that. Well, we care because aneuploidy in human preimplantation embryos is exceedingly common. And of course, as we all know, it's a problem that increases with advancing reproductive age. And here's that sort of famous kind of shape of graph that you get with incidence of Down syndrome or miscarriage or anything to do with chromosome problems with advancing age. What it shows us is that even in the young patients, aneuploidy, and this is in blastocyst stage embryos, so embryos that we'd usually consider to be quite nice embryos, they've made blastocysts, that's good. Even in the young patients, these will typically be egg donor ages, we're seeing an aneuploidy rate of over 25%. So 1 blastocyst out of every 4 being chromosomally abnormal. But the scary thing is that beyond the age of 35, as you see, it goes up very dramatically. So for a patient over 40, you're typically talking about 3/4 or more of the blastocysts being chromosomally abnormal. And in fact, beyond the age of 35, the aneuploidy rate actually is increasing at about 0.5% per month. So really every month counts, particularly for patients who are already in the late 30s or over 40. Of course, the impact of that is that these aneuploidies are almost invariably lethal. We've known for decades, of course, that aneuploidy is the principal cause of miscarriage, causing at least 2/3 of miscarriages, but likely more. But actually it also turns out to be the principal cause of implantation failure. When you transfer a nice looking embryo in an IVF cycle and you don't get a pregnancy, the most likely explanation for that is that it was chromosomally abnormal. So not surprisingly, as aneuploidy rate goes up with age, the implantation rate goes down, and it's no coincidence that these two lines are almost a mirror image of each other. What you're seeing is cause and effect. So, unfortunately, our regular PGD protocols, nice as they were, did not detect aneuploidy, and so we're potentially transferring embryos that are going to end in a miscarriage. And in fact, we've seen this many times in PGD. You get a couple, they're desperate to have a child, you're delighted because you find a morphologically perfect, normal embryo, and you transfer it, and they get pregnant, everyone's delighted, and then you hear a couple of months later it ended with a chromosomally abnormal miscarriage because you hadn't checked that. So it, of course, would be nice to be able to see these aneuploidies as well. It would also be good to have a more generic test, something with less customization to hopefully bring down the workup time and maybe bring down the costs as well. And ultimately, these have been now realized with the introduction of karyomapping, which I guess we've been using for about 5 years now in Oxford. It's based on looking at about 300,000 single nucleotide polymorphisms scattered throughout the genome using a Illumina bead array that looks something like this. But essentially what we're doing is looking at a kind of DNA fingerprint for each region of the genome. And of course, we're going to focus on the gene which is causing the problem in the individual family. So we'll look at polymorphisms across the entire genome, but then we'll focus in on the ones around our gene of interest. So imagine a mother and a father, 2 copies of each autosome. Maybe the mother is carrying a mutation on her blue chromosome here. Rather than design a patient-specific test looking only at that mutation, we're going to essentially ignore the fact that the mutation is there and instead look at polymorphisms that are either side of the mutant gene. So some polymorphisms above and some below, and we're going to type her. So she's homozygous for the first one, AA, then heterozygous, and so on. We'll also type her partner, and then hopefully there'll be another individual in the family who also carries the same mutation, maybe a child, for example, and we'll type them too. So we have this genetic information, which we're then going to track through the family when it comes to their embryos, and maybe we'll get an embryo with a result like this. Okay, at the first site, we're only detecting A, so presumably that means it's homozygous, A on each chromosome. Heterozygous at the next two sites, and so on. Of course, the question is, what does that really mean to us? Well, what we're looking for is evidence of the inheritance of individual chromosomes from the parents. So, for example, a B allele at this third position and an A one at the penultimate position is only seen on the father's green chromosome. So that chromosome must be present. Similarly, an A at this second position and a B at the last position is only seen on the mother and the affected child's blue chromosome. And we know that that's the one carrying the mutation. So without ever having looked at the mutation itself, we know that the mutation must be there, and therefore this embryo, if it's a dominant disorder, would be affected. You can also get results like this. Here again we have evidence for the father's green chromosome. Happily, this time we have evidence for the mother's pink chromosome, so the embryo should not be affected. But unfortunately, we also have data suggesting that the father's yellow chromosome is present. So while this embryo is unaffected by the inherited condition, it is chromosomally abnormal, and therefore we would not want to transfer that embryo. I can show you a bit more what this actually looks like as presented to us by the software that does this analysis for us. We don't actually sit there, of course, doing this manually. Here you have a sort of cartoon that the software generates. Blue and red for the father's chromosomes, sort of gold and green for the mother's. And then here's a result for one chromosome. Each one of these little spots is essentially the result of a SNP, a single nucleotide polymorphism, which is confirming the presence of, in this case, the father's blue chromosome. And the key thing here is, okay, they've got lots of pieces of evidence all telling us that the father's blue chromosome is here, but when it comes to the mother's chromosome, it's not detecting the yellow or the green. In other words, this embryo actually has a monosomy for this chromosome. We can visualize it in other ways as well. This output basically shows the amount of DNA that's hybridized to the microarray. It gives you a kind of quantitative measure, and I think you can see straight away over there that one of the chromosomes actually has less DNA on the probes that are associated with it. So again, this is showing a monosomy for chromosome, in this case 18. We can also look at that in yet another way. This is showing us the actual genotypes at each of the polymorphisms. Each one of these spots corresponds to one of these 300,000 polymorphisms, and they've just been put in the order they appear along the chromosomes. And what essentially you're seeing is one kind of homozygote. These are all the spots at the bottom. Another kind of homozygote is all the spots at the top, and the heterozygotes are these ones scattered in the middle. And you can probably see straight away, when we look at chromosome 18, it doesn't have any heterozygous loci. And again, that's because there's only one copy of the chromosome there, so it cannot be heterozygous. So all of this data can give you some nice information on aneuploidy. So how's this changed things? Well, the workup time can be as little as about 48 hours, although realistically it's usually 2 or 3 weeks to get this done, but certainly way less than it used to be with conventional methodologies. You're detecting most aneuploidies this way, not all of them. There are some that creep through undetected, but you'll detect virtually all the meiotic ones, which are probably of greatest concern for us. Of course, mix-ups almost never should happen in a modern IVF clinic, but if there ever was a mix-up of sperm and eggs or embryos from the wrong patients, you would immediately detect it here, because you would see that there was a problem with the genetic relatedness of the embryos to their parents. You don't really pick up incidental findings. That's been people have asked in the past, well, you're looking across the whole genome, so don't you pick up incidental findings that maybe you weren't looking for? The answer is no, really, because the resolution of this is not sufficient to do that, and also the polymorphisms we're looking at are not necessarily related to any diseases. Just a little bit of clinical data from one clinic that uses this service a lot with us, about 200 cases they've done in the last couple of years. Their implantation rate is actually 70%. If you compare that to the published PGD implantation rates from the ESHRE PGD consortium, it's actually 3 times higher than that. It's hard to know exactly why that is, but I would suggest it's probably largely because of adding in chromosome analysis into this picture. We're not transferring embryos which ultimately aren't going to implant or will miscarry due to a chromosome abnormality. The loss rate, again, very low. Only about 7% of pregnancies actually fail to go to term. And that, again, that's much less than you would expect. I have to say this is not controlled data in any way, it's just observational, but I think that's interesting and probably indicative. 63% of cycles gave a birth per transfer, or 68% if you're only looking at the blastocyst biopsy cases. So that looks pretty reassuring. A couple of publications on this recently, if you're interested. So that's where we stand today. Karyomapping has been really transformative for us on the PGD side of things, and has been a real welcome addition to the arsenal that we have available to us. It's great to have Graham Snudden in the audience because he was one of the founders of Bluenome, which was the company that really managed to deliver this to us. It's an idea from Alan Handyside, but delivered by Bluenome, which was one of our favourite companies. So just to finish up really on where we're headed next, well, I'm sure Joris will talk about next generation sequencing. I'll just briefly mention how it's being used in a more general context. And you know, I guess a lot of you have a genetics background, but perhaps not all of you in the audience. And so I'll just mention that really next generation sequencing isn't one technique. It's an umbrella term for a number of different technologies, which I guess all have in common the same thing, that they deliver a very large amount of DNA sequence information at low cost and relatively quickly. Essentially what we're doing in the PGD context is we're taking the DNA out of the biopsy sample. We're doing a whole genome amplification to try and amplify all of the DNA in there, or at least as much as we can. Then it's going into the usual next generation sequencing pipeline where that DNA is fragmented into pieces, and each one of those fragments is sequenced. So we learn the order of the genetic letters along the length of literally millions of short pieces of DNA, usually referred to as reads. We're really only using this for chromosome copy number analysis at the moment. So what happens is we're faced with this huge jumble of millions of sequences. To you or I, it's meaningless, but to a computer, of course, it can align these to the known sequence of the human genome, tell us where in the human genome that little piece of DNA matches, where it came from. And of course, that tells us then which chromosome it was from. And so you end up with literally thousands and thousands of fragments of DNA from each chromosome. And you can then say, well, if there's 2 copies, we expect a certain number of fragments. But if we detect too many, that's because the embryo has an extra copy of that chromosome. Or if we detect too few, that's because the embryo didn't have enough copies of that chromosome. So this works very nicely in the context of PGS. But in terms of PGD, what I've been talking about during this talk, of course, we need to get down to the actual DNA sequence information. Now, for the way that we use next generation sequencing in PGS, we sequence less than 0.1% of the entire genome. And actually, we've been very happy with that because we don't want to stumble across incidental findings, to do other diagnoses of genes that hadn't been requested by the parents. We don't want to get into that ethical sort of quagmire of finding things that we maybe don't even understand. So we've been quite happy to have this low resolution, just counting the copy number of the chromosomes. But ultimately, of course, things are moving in that direction. And there could be some advantages maybe if we did have more detailed sequence information. We would then have a methodology that could be applicable to any disorder. If we think about how PGD and how PGS have evolved over the years, they've never really overlapped. At the beginning there was FISH for looking at chromosomes, and there was PCR and then multiplex PCR for looking at genes and sequences. Then we got a bit better at doing the chromosome analysis. We brought in metaphase CGH, which was rapidly replaced by array CGH. Karyomapping came in, and then we've got the next generation sequencing. But really these two methodologies are targeted at different questions and don't overlap that much. The possibility is that if you did do whole genome sequencing of embryos, you would have a single protocol that delivered both things simultaneously. So that could be desirable. It might in improve embryo selection. We would learn about the chromosome copy number as well as any gene disorders. We'd be able to quantify the mitochondrial DNA, which now seems to be relevant for embryo viability. We might detect gene mutations in the embryos that affect their viability. Even if you transfer chromosomally normal, morphologically perfect blastocysts, still about a third of them do not make a baby. Why not? Some of it may be endometrial, of course, but I think most people think that may only account for 5 or 10% of failures. So what's the rest of it? Presumably embryonic. Are there dominant mutations that occur de novo, which cause problems in embryos? Are there inherited recessive mutations that are preventing some of them from implanting? Of course, if we had the whole genome, we could look at both the mitochondrial DNA, the nuclear DNA, and get that full picture. Of course, it's also problematic though, because it may reveal mutations that are relevant to the health of the infant. Is that problematic or not? I don't know. Certainly we're likely to come across unexpected findings that will may or may not have implications for health, and so counselling becomes a little bit nightmarish. So we have to think very carefully whether this is going to be acceptable or not. How would you counsel patients about the unknowns, or maybe how do you choose between an embryo which has a 10% increase in its risk of breast cancer versus one that has a 15% increase in the risk of cardiovascular disease? It's going to get very complicated. But the reason I mention it is just simply because we're approaching a point where it may be possible to do this. In the past, as a geneticist working in this area, it was always very easy just to wave your hands and say, oh, well, that's technically impossible. Or it costs too much to do. But of course, the cost of sequencing has gone down so rapidly. I remember when the human genome was first sequenced, it took over a decade and cost more than $1 billion. But you can sequence a human genome now in about a day for about $1,000. So these bars, both technical and economic, to doing whole genome sequence analysis of embryos are starting to fade into the background. So we really do have to think about these questions sooner rather than later. So I'm going to leave it there just to acknowledge that some of the clinical data I showed was generated by our group here in Oxford in collaboration with Reprogenetics. Got a really great group there now doing some fantastic work, so I just wanted to mention them. And thank you for listening.